Now, I have been difficult to catch over the first 3 days because I shot myself in the foot. I'm not going to go into details, but just realise that for every minute of my talk, and I'm wasting them right now, 20 minutes, I spent 4 hours getting here. So that was 80 hours, and I've been flat on my back, asleep since then, practically. So, all right, let's go. This presentation follows on in many ways from the talk that you've just had from Paolo, and it asks the scientific question, is there a biological reason for the coloration of shroud-like linen that was found using the lasers, as explained by Paolo and others in 2 papers that they published in 2010 and 2012. Now, if we suppose the shroud authentic, we can also ask, is there a connection between the coloration of the shroud, of the image found on the shroud, and the biophysics within the shroud man's body? Another way of asking this question is, could the many chromosomes within shroud man's body have acted as tiny lasers? And we've seen that being used a few times before. So we're talking about many little loop antennas all over the body emitting UV light perpendicularly outwards from within shroud man's skin and thus forming the image we see on the shroud. This present study looks at the possibility that shroud man's chromosomes may have reacted with the shroud since the wavelength of both are very close. The chromosomes in shroud man's body and the argon fluoride eczema laser, the lasers that were 193 nanometers that we have just discussed. In the original studies that Paolo and others from ENEA found that exposure of shroud-like linen by a single argon fluoride eczema laser operating at 193 nanometers could accurately simulate the coloration found on the Shroud of Turin. These tests also showed that the superficial nature found on the shroud corresponded to a fair degree to the laser irradiation of shroud-like linen. According to Professor Giulio Fanti, more testing is necessary before affirming that the superficiality of only the primary cell wall was obtained using laser irradiation. It was realised biologically that 30 nanometer chromatid fibre, a well-known standard of cell biology, also resonates at 188 nanometers, very close to the laser operating at 193 nanometers. The difference between the studies, the argon fluoride physical laser and the many biological nanolasers, is that instead of one argon fluoride physical laser and the many biological nanolaser, is that instead of one laser, the body has 23 chromosomes per skin cell of around 30 micron of size. In other words, the body has 2.3 by 10 to the 10 chromosomes per square metre. It may be too expensive to do a physical experiment using nanolasers, to put it mildly, but even over a limited region such as the face, but it may be possible to do computational testing in the future. I think Mario spoke about computational simulations a couple of talks ago. Now, chromosomes are known to have UV flows down their axis during metaphase. Each is made up of 2 chromatids, and they rupture at metaphase into 2 daughter chromatids, each one having 1 single spiral set of base pairs. And there are 3 billion base pairs across the full set of chromosomes in each cell. So we're talking about their fractal structure is currently, as the slide is saying, there's more than some, there's many unknowns about the chromatids and how they operate. If we come to the cell cycle, it's been gathered together by various researchers for about the last 200 years. And it's basically known to be a series of stages that achieve overall replication of a single cell into 2 cells. Metaphase, the most important phase as far as the image on the shroud is concerned, is accompanied by flows of UV down the axis of the chromosomes. These are endogenous fields, in other words, they are coming from inside the body and not from outside the body. The cell cycle incorporates several different physical methods by which these endogenous fields are created, including diffusion of dipolar proteins that float around the plasma membrane of the cells. There are various resonances and other more specialised biophysical reactions. Endogenous fields are generated also by colonies of cells, not just a single cell. And again, it's the dipolar proteins that are moving around within the plasma membranes of all cells in the colony. There can be normally a stimulus, 1 or 2 cells in the centre of the colony, which is already charged on its surface by the motion of those proteins that I'm talking about, these diffusional proteins. In the case of the shroud, it appears that something different is happening. It's important to note that the cell structure is also fractal, the same as the chromosomes. So the endogenous fields are ranging across these fractal structures, both the cell and the chromosome. The endogenous fields that are coming from within the body range over these fractal structures of the biological tissues from quasi-static fields across a whole colony of cells down to the tiny size of the base pairs of the DNA within a single cell. And once it's done that, once it's ranged across that frequency range, it starts again. As discussed by Paolo, if shroud-like linen could be colorised similar to that of the shroud, this was an ageing without burning of the primary cell wall of the flax of the linen. That is coming from a study published in 2012, that wording, ageing without burning, quote unquote, of the primary cell wall. This work today also is compatible with my previous hypothesis of biophotons coming down the chromosomes in 2014 that I presented in St. Louis. Now, in a short time ago, I realised, and as you see down in the bottom right-hand corner there, you can see lambda equals 2 pi d. The lambda is the 193 nanometres. Well, in this case, it's 188 nanometres because the d is 30 nanometers. So you take 2 pi or 6.28, whatever it might be, and you end up with 188 nanometers. So that's the basis of this whole talk, that there's a very close relationship between the frequencies that this is suggesting and the argon fluoride excimer laser. Now, this is some other work that I've been presenting recently. It's a combination of the cell cycle as a field phenomenon inside the body, as well as biofields, I should call them, as well as a biochemical view. And so what we see in the top right-hand corner, I'm trying to find the laser pointer. Where is it here? Ah, there we go. That part of the slide there in the top left-hand corner is a dispersion characteristic, the permittivity of biological tissue. And we can see various levels and drops, like a snake and ladder type of thing, but it's actually sloped so that it's acting like gravity, so that it sort of starts off up here and then it keeps on rolling and falling, and it goes into these different fractal parts of the cycle that I was talking about previously. The stages can also be seen as regions within a colony of various grey colours on this little slide here that show like a little pimple. And what's happening in the centre is that we're getting a superposition of electromagnetic fields. So this is a view on top of all the genetics and biochemistry that's involved. This is the big picture. The chemistry and the genetics is a much smaller picture, and we'll come to that. So this is what happens at metaphase. We get this alignment. These are meant to be chromosomes here. So we get an alignment across what's called the metaphase equator. And we get a little flow of UV along that axis. The difference between our bodies and perhaps Christ's body is that we only do this very infrequently. Our body's going to metaphase when it's required. Maybe we've had an injury or something and the body will go into overdrive at that point, and it'll go through mitosis and start regenerating more cells. So that's what's happening here. But it's important to note that we actually know this mathematically pretty well. Where the parts of this cell cycle we do know very, very well mathematically and from a field point of view. These locations of these chromosomes are mathematical. We'll come to that, I think, maybe the next slide. This is a waveguide of a mode, a TM11 mode of a circular waveguide. Now, the reason I'm using that is because I don't have a picture for an ellipsoid or a three-dimensional structure like a cell, an axisymmetric cell structure. So, but it is, in fact, very... It's very close to the shape that we've got there, and you can see some similarity between that and that. If we go to the next slide, first of all, I'm going to talk about the diffusions that are happening at the lower frequencies. what happens, quite amazingly, this is really quite amazing, the cell becomes very round and very stiff and tensile. So even though I've shown them as little spheres or axisymmetric shapes, in fact, they're pretty much like that. Because to generate all the electromagnetic fields, the only way they can do that is by tiny little spherical rotations, tiny little circular rotations of these dipolar proteins that are part of the membrane. And because they are on a three-dimensional surface and you've got little circles, you actually get electric, magnetic and vibrational fields all in one. They eventually cause things like you see at the bottom there, so that there's a partition of the various charged dipolar membranes. I've just made a note here that we've seen various times where the comment was that the cloth is flattened. That's been a phrase that one or two people have used this afternoon, and one of the reasons could be that, the cells, all cells, including the skin, gets rounded and stiff, so that this could introduce some of the features we're seeing on the shroud. Okay, here we go. The big picture. This also fits in with some comments that I've seen today. Just let me go on to what I was saying just a minute ago about the cells becoming very round, very stiff, very tensile. So it's not just gravity that explains the image, as Bob Rucker and others have spoken about. So we've spoken about gravity. These are not just mathematical little ways of representing the cell. They are various higher wave modes. And if we juxtapose our thinking and thinking about what's happening to life, is there another cell cycle going on here within Christ's body? Perhaps life can be found to exist at higher energies, as Jose Godinez suggested earlier today. Okay, so again, this is describing what happens. Eventually, the chromosomes break and they become two little chromatids. One chromatid goes up to the north, another one goes down below. And at metaphase, they're getting there. So the whole thing is being stretched into two bits, one north and one south, and then eventually it breaks. So we're talking about some sort of, it's called a kinetochore, and the kinetochore is a part of the linkage between the two chromatids that are joined together in the chromosome. Each chromosome, you remember we've got 23 of these. And this is the physical effect. This is the well-known Stark effect on the centromere. So as a quasi-static field builds up, and that's building up because there's a diffusion going on where the cells are separating into positive and negative, and so some of the cells are undergoing a a build-up of quasi-electrostatic fields, and the centromere bond is getting stretched and eventually breaks. Here's a schematic that we can guesstimate. This might be of interest to Mario again. Where are you? I can't see you. You might be interested in this, Mario. This is my idea out of my brain. So we have, oh, sorry, we'll come back to that. Wrong one. So we have 30 nanometres. So I've schematically put a chromosome there and a chromosome there, which we've seen before. There's actually like 23 of these all aligned, all behind one another in a particular skin cell. There's another 23 up here. So this is Shroud man's skin, and this is the flax. This is the linen shroud, and this is the each flax fibre. We've got a primary cell wall, which is 200 nanometres roughly across, and we've got a secondary cell in there. So what I suggest is happening is that we've got a wavelength of 188 nanometres coming here, UV, and it's coming across this 200 nanometre, I call it a lossy fibre, because it's a mismatch of frequencies. It's going to cause a power heating of some kind. And so, again, you can see, you can see there are bits of this that are corresponding with other people's work. Okay, this is perhaps the nitty-gritties of the talk. What we got here is a schematic of the skin in terms of its cells, skin cells. And although it looks pretty curvy, each of these is 30 nanometres in diameter or in size. So you can see that the UV is pointing pretty much perpendicularly out. So I, like Mario, think that a vertically collimated UV field is not what's happening. I think it's perpendicular to the body everywhere. You've got to remember that there are 23 billion skin cells per metre squared, so there's quite a lot of them. Finally, the magnification that can be achieved by one chromosome to another chromosome is very much like the coils in a transformer, where the magnification level is by taking the number of coils in the primary and the secondary and multiplying them together. So that's exactly the same here. And we don't need many. We only need one centromere, which is the central linkage between the two chromosome, between the two chromatids. If you take the 23 and put them in the terms of 2 to the 23rd, you get a magnification of 8.3 by 10 to the 6th. Oh, sorry. Keep on pressing the... So this corresponds to the centromeres rupturing. So 2 or 3 cells can give a large amplification. I think that's saying 7.8 by 10 to the 16 photons, which is what I worked out was the quote-unquote peak effective laser amplification for a 193 argon fluoride excimer laser. The difference to normal biology is that all the cells of Shroud man's body need to be in something like mitosis. As I said to you before, it's very rare for our cells to be in mitosis. It only happens when we need it. Excuse me. The antenna equations, which we can apply to this, imply that the chromosomes may form an array of 8 by 8 cells in size. And the lossy fibre may form a natural filter on fluents, avoiding any ablations. So finally, overall, the biological evidence strongly suggests chromosomes act as antennas and transformers, and thus may have played a role in the image formation. And that is part of another ongoing area of work that I'm involved with. The two wavelengths, human chromosome and argon fluoride excimer laser, are accurate to about 97.5%. The final point is that does a version of the biological cell cycle underlying what happened to Shroud man during resurrection? And that also raises a number of other intriguing questions. Thank you. Sorry?
Do you have time for one question?
Yeah?
Time for one question?
Yeah.
What's the energy source of your antenna?
Excuse me. There's a question.
Pretty low. It's about 1,000 photons per second. So to go from that to the argon fluoride excimer laser